Hello. Today I'm going to talk about the evolution and genetics of hybrid lethality in Mimulus. And so what I really was trying to do at the beginning of my dissertation is answer this question. How are reproductive barriers maintained in the face of ongoing gene flow? Which is a really important question. And to answer that question to some extent, I've been studying Dobzhansky-Muller incompatibilities. So just to go over this, these incompatibilities cause hybrid dysfunction when they occur. The way it works is that traditionally, like the classic, is that you have an A gene and a B gene. These interact somehow with one another. And they could evolve if you have a little B allele evolving on this divergent lineage in the big A background, and maybe a little A allele evolving on a big B background over here. When these two species come together and hybridize, little a and little b, which have no co-evolutionary history with one another, now are interacting, and that interaction might be all screwed up. And so you wind up with unfit F1s and F2s. So there are some big questions regarding Dobzhansky-Muller incompatibilities, like what are the types of genes that underlie incompatibilities, the genetic mechanisms that lead to their evolution, and also, how in the world are they stabilized when species hybridize? So I've been studying these in the Mimulus guttatus complex in these two species. Mimulus guttatus, the large outcrosser. Misutus is its sister taxa, it's a sulfur. They have a broadly overlapping distribution with Misutus nested within guttatus. They're isolated by a lot of pre- and post-zygotic barriers, but despite all of this, there is ongoing hybridization at a pretty low rate within the system, with introgression largely going from Mimulus misutus to Mimulus guttatus. Now, if you cross certain lines of Mimulus guttatus and misutus reciprocally, in the F2 generation, you wind up with all these white seedlings. They don't have chlorophyll, and they die shortly after germination, which is pretty crazy. As it turns out, it's 1/16 of the progeny die, and early on I mapped the loci involved. So these are, I call them hybrid lethal 13 and 14. So these are like loci here for Misutus and guttatus. Guttatus always yellow, Misutus always blue. And if you're homozygous for guttatus alleles at 13, at that locus on chromosome 13, and homozygous for Misutus alleles at 14, you always die. This is a completely lethal genotype, and that defines what causes white seedlings. Recently, and this is like the biggest, this is like really cool, I actually found the genes that underlie this and the mechanism. So gene duplication underlies this incompatibility. This is called PTAC14, and the kind of model, it's a hypothesis right now, is that ancestrally you had a copy of this key photosynthetic gene called PTAC14 at hybrid lethal 13. In the guttatus lineage, this gene transposed to 14, so now you have two copies, probably relaxing selection on one of these copies. This copy here acquired a premature stop codon and doesn't make any transcripts at all, and so you're left with a bad copy here. Nothing changed in the pseudo, so you just have no copy here. So if you're homozygous in the F2 for the bad copy and no copy, you don't have this critical photosynthetic gene and you die. Now, doing all this genetics allowed me to start investigating what I really wanted to do, which was study incompatibilities, a specific incompatibility in the wild, which has really never not been done very much. And so I want to study the evolutionary dynamics. How does little a increase in frequency or little b? So, and what's going on at this hybrid zone barrier? And we can, ascertain some of this information by looking at populations, allele frequencies in populations. We can answer really basic questions. Are incompatibilities allele fixed or polymorphic? What's their distribution? Do they actually occur? Are they expressed in nature? And how are they maintained when you have gene flow? So here is the distribution map for hybrid lethal 13, the one that's bad with guttatus. Yellow circles are guttatus populations, blue are misutus, white is the incompatible allele, and green is the compatible allele, so that one's okay in the cross. And what you can generally see is this incompatibility allele is very widespread and polymorphic within the system. These are global allele frequencies, so you can see that Gutatus generally has a high frequency of the HL13 incompatibility. Nasutus has a different low frequency. If we look at the other allele, you see it essentially the opposite pattern. A high frequency of incompatibility in Nasutus and low in Gutatus. So these are the global allele frequencies. And what we're interested in, what is the source of polymorphism at these incompatibility alleles? And there's a couple non-mutually exclusive hypotheses that I'm starting to get at with my data. One is introgression. You could imagine if the incompatible allele here is ancestral, and there's unidirectional gene flow from Nasutus to Gutatus, that maybe these good alleles are just introgressing in this direction, and perhaps here too. So one hypothesis is that there's gene flow that's contributing to polymorphism. The other is that there's actually a pool of ancestral variation, that these things, the ancestor actually had variation at these alleles already, and so you have incomplete lineage sorting that's still going on within each taxa. And you can start to answer which one of these it is by looking at haplotype variation or genetic variation in the region surrounding incompatibility loci. So like I said, some Gutatus individuals have a compatible hybrid lethal or 14 allele. And so if it's got a compatible allele, you just genotype a couple markers flanking the incompatibility region, and you just take the size of marker 1, marker 2, and just call that a haplotype. So I'm using like 2 locus haplotype stuff. You can do the same thing for incompatible alleles, and you do that for Nasutus and Gutatus. So you partition Gutatus into compatible and incompatible types and look at their haplotype structure. There's a prediction to be made about haplotype structure. This is the decay of linkage disequilibrium in Gutatus and Nasutus. So LD decays really slowly in Nasutus. It's a sulfur, so you get this maintenance of haplotype structure. So you might, in Nasutus effect, expect, if this is all the haplotypes in Nasutus at some locus, maybe just 2 haplotypes. But LD breaks down really quickly in Gutatus, so you might expect like a whole bunch of haplotypes in Gutatus. Now, we can test our introgression hypothesis. We're testing this hypothesis. Are these white alleles the same as those white alleles? And so if we look at Nasutus, white guys, and Gutatus white guys, and if they have the same haplotype structure, then you might infer that there's introgression going from Nasutus to Gutatus. Alternatively, some process of incomplete lineage sorting might show an opposite pattern, where you have just this incompatible alleles have existed in Gutatus for a long time, and so the haplotype structure looks very similar between compatible and incompatible Gutatus alleles, and that variation around the incompatibility. And we have very clear predictions about what we expect. So this is the incompatibility. We just changed the names to good and bad. So if you're in Nasutus and you're introgressing that way, you can bring this good allele and move it in and replace that bad allele. And what the result is that over time, that good allele should just replace the bad allele, and you can have all green seedlings and purge the incompatibility from the system, which is what you might expect. At the other locus, this bad allele going in here, you don't expect that to happen. Introgression shouldn't happen at that locus because it creates lots of bad copies, lots of white seedlings, and selection would not, it's just not going to allow for that. It's not likely at all. So here's the actual data. This is testing this one, the good allele going into the bad, replacing bad. So here's the haplotype variation that exists in Nasutus individuals for the good allele. This is the haplotype structure for good individuals of Gutatus with the good allele, and these are the Gutatus with the bad copy, which we think is the ancestral copy. And what you can see is there's quite a bit of overlap in the haplotype structure here and here, indicative of gene flow from Nasutus to Gutatus at that locus. And that suggests that the incompatibility itself might be being degraded. Moving on to the other incompatibility locus, which is bad going into good, we get this surprising result. So this is a bad copy. This is, in Mimulus nasutus, every single individual has the same haplotype around this bad allele. Essentially, every individual from Gutatus, the other species, also has that same haplotype structure. So this doesn't make any sense at all, like zero sense. So not based on like the predictions I gave you earlier. So I have like a different, hypothesis now. So my thought is that probably there is hybridization, and this allele actually is getting into Gutatus, but these Gutatus individuals are very likely early generation hybrids. So it's a recessive allele, it can at least get in there in the heterozygous state. But after a couple generations, when recombination's happening, essentially you expose that incompatibility allele, and selection's just gonna knock it back right away. That's what I think is happening. But I don't have, it's a little inconclusive. So I'm trying to figure that out right now. In any case, it's a lot of interesting patterns about what might be happening in Mimulus with this particular incompatibility. So, in conclusion, gene duplication underlies hybrid lethality in Mimulus. Hybrid lethality alleles are widespread and polymorphic. There's definitely evidence that hybridization contributes to that polymorphism. And hybrid incompatibilities might be being degraded in the system, but we're not entirely sure yet. It'd be interesting to know if they might be. And with that, thanks.
I may be reading through the lines, but it sounds like the assumption is that for the having two copies, two functional copies, one of each locus, is you're considering it to be equally fit to having only one. But what would happen if, in fact, there's a deleterious consequence of having a functional locus at both, functional allele at both loci?
Yeah.
I think you've looked at the fitness of that combination relative to the wild type.
I've never done that. It would be interesting because, it would be really, really fascinating to challenge the fitness of this bad copy to this good copy. That would be one thing. And to see and also two good copies in this particular individual. So two good copies could have the potential to just lead to like less fitness, right? That's what you're saying?
Yeah, they have a balancing effect.
Yeah, and I've never done that. I think that would be interesting. So we kind of expect the incompatibility to just be degraded because this particular incompatibility allele is just dead. So you're really interested in the fitness of this bad allele compared to this good allele in this genetic background mostly. It's difficult to know, but it's a good question and very interesting.